Robot Talk is the podcast that sits down with robot enthusiasts from around the world and asks them the questions you always wanted answered. Like, how do you make a microrobot? And how does that thing work? Hello everyone! Welcome to Robot Talk. I'm your host, Claire Asher, and this week we're zooming in to the microscopic world of nano and microrobots and finding out how these tiny devices are set to revolutionise medicine and scientific research. Before that, please remember to like or rate this episode and leave us a review if that's something you can do on the platform you're listening on. And don't forget to share the podcast on social media or tell your friends about it. It really helps us reach more listeners, so thank you in advance. This Sunday, I'll be at the V&A Museum in London for Robot Talk Live, where I'll be chatting to 3 awesome panellists about robotic prosthetics and human augmentation as part of the Great Exhibition Road Festival. If you'd like to join the audience, there's still time to register for a ticket using the link in the episode description. Hopefully see you there. So, with all that said, it's time for me to introduce this week's guest. I recently had a great chat with Simone Schuerle from ETH Zurich, all about microrobots, medicine, and science. This week, I'm talking to Simone Schuerle, an assistant professor at ETH Zurich, where she leads the Responsive Biomedical Systems Lab, developing diagnostic and therapeutic systems at the nano and microscale. Hi Simone, welcome to the podcast.
Hi Claire, thank you so much for having me.
So, nano and microrobotics is a topic that we haven't really covered that much on the podcast before, so I'm excited to have you on to talk about it. So what are some of the biggest challenges of building robots at this kind of scale?
Well, several. To start off with, basically, their fabrication, right? So there are kind of 2 main streams. You can go top down, as we say, where you break up bulk material, make it tiny, and then put components together through assembly techniques. Or you work bottom up from basically, individual building blocks, a lot of chemistry. And these are kind of the 2 angles you can take, right? But no matter which route you take, it's really a convergence of sciences. So there's always lots of chemistry involved, physics, different physical principles. For example, self-assembly processes, how you can make use of surface tension or charge effects, hydrophobicity, and so on. And then, of course, engineering principles. So basically finding ways to effectively fabricate at the scale and then at high throughput, because very often we need actually many micro or nano robots to be effective in what we want to do. But I mean, there's a plethora of technologies basically now out and approaches and strategies. And we can make use, for example, of microfluidics, where we create micro channels where we can feed in different components, like, for example, hydrogels that we can use as a material, like basically polymers, and then magnetic nanoparticles that I work a lot with to make these little robots responsive to magnetic actuation, for example. And it can then, by having an aqueous and oil phase, you can basically create little droplets, and then you can, by UV exposure, polymerize them and so on. So there are different techniques. This is just one of many.
Mm-hmm. Yeah, it strikes me how different this is than, the process by which you make a large robot, the kind of the engineering things that I'm talking to people on the podcast normally about.
Yeah, it's actually such an important point you're making. I mean, we should really take also a step back and ask ourselves maybe, how does a microrobot look and function, right?
Yeah.
And it's really not that we should imagine like a macroscopic robot as we would know them also from movies or even in our daily lives, that basically it's not scaled down versions of that and tiny little metallic boxes with little gears and motors, because actually there's a lot that doesn't work very well in scaling. But why would we call, for example, such a hydrogel droplet with magnetic nanoparticles a robot, you might ask, right? And to me, I mean, they still follow a very fundamental principle in robotics that is basically you have a system that there is a certain external input cue that could be magnetic field, light, heat, whatever, a trigger. And then there is a processing rule applied. So the system is in some way responding, and then there's a certain distinct output. And that's kind of then what we're working with by knowing and using materials that respond in a certain way to a certain input. And the output could then be that there is a swelling and a release of a drug, or or directing motion. I mean, we can direct motion with this by responding to external magnetic fields. We can pull and drag. We can apply rotational fields. We can start having them tumble and roll, and like that's how we can create motion. Which also comes to the other challenges of how can we power them? What are effective ways of powering them? Because we can't really put batteries on them. It's very also inefficient at scale. And so here we work, for example, a lot with magnetic fields and magnetic materials and a way to transmit that energy effectively wirelessly, right? Because they need to be, in that sense, untethered, autonomous. And so, there are different approaches. People have also worked with ultrasound as a way to manipulate. So working with acoustic waves, but also with light or other cues. Some also make use of basically chemical reactions that can be used for propulsions and catalytic reactions.
Yeah, that's really interesting. I guess the other thing, I mean, the main area I've seen this kind of technology being applied to or being suggested that it might be applied to is working inside the human body. So you have to make sure that all of these things are safe, biocompatible, not going to harm people.
That's true. I mean, that is one big application area. But I think before we enter on that and all the challenges associated with that, there's really also a huge application space outside the human body to, for example, study single cells, still in the context of life sciences, but having basically means to manipulate on a micro scale, right, with objects, of living organisms, with, for example, in one study I actually did during my PhD, I was trying to probe the forces of macrophages, our immune cells, how they attack and uptake prey, basically, for example, bacteria. And so we had a little, in the end, just a magnetic bead that we could functionalize with similar antibodies that they would also encounter for a bacterium and kind of mimic, it also had a similar shape to kind of the interaction and learn about the forces they apply and the tactics. And this is basically here, such microrobots. In the end, I mean, there's a lot of robotics in the control instrumentation, right? In the magnetic system that generates the fields and also gradients to basically maneuver these tiny magnetic objects. But this is a way of, basically, for example, studying cells, but also now at a tissue level, there's actually huge interest also in trying to understand how muscles grow, or if you try to make little artificial tissue that also includes living cells, right? Having means of basically applying forces that they would be also exposed to in the body, and so on. So there's a lot where microrobotics comes in to understand at a cell and tissue level. Also for certain micromanipulation tasks where you have, hard to reach environments. This could be actually helping in little chemical reactors to have little robots to just bring in some convection by having them and swirling around. Like these are, some other examples. I mean, a field I'm really excited about is, of course, their use as diagnostic and therapeutic microrobots in the human body. And that's the major focus of my research also.
Yeah, it's a fascinating mixture of physics, chemistry, engineering, biology. It must be a really exciting field to work in. How new is this field of research?
That's a great question. I mean, I think it goes back to basically early 2000, where we heard about the first ideas and concepts of microrobots and designs. And it's been interesting to see. So a lot of the microrobots that have been developed are actually also inspired by nature, or bio-inspired, as we say. So as it's very difficult to move at a small scale, there have been mechanisms, for example, how bacteria move forwards, being copied. So they have these nice flagella and a molecular motor that spins very nicely, and that translates them forward and moves them forward like a corkscrew. This corkscrew propulsion. And that has been, for example, being copied by making artificial flagella, like basically little helices that can be turned with a rotational magnetic field. So if they're magnetic, then they would also spin, but you don't need to build a complex motor. And so, there have been lots of creative solutions and a lot of also inspiration from nature.
Yeah, that's really interesting. So as you mentioned, one area you're currently working on is micro and nanorobotics for drug delivery. So can you tell me a bit more about this work?
Yes. So, I mean, a major bottleneck today and generally in treating diseases is getting drugs effectively to the target site where they're actually needed, right? And so a lot of the phase 3 clinical trials also fail because of insufficient efficacy. Like the dosage you need to basically show efficacy is just then too high, would have too strong side effects and basically be toxic, right? So we need to think about ways how we can more effectively bring what we, for example, intravenously bring into the body to a target site. So in particular in cancer therapy, for example, still one of the most commonly used therapeutic strategies is chemotherapy, where you would, have an ongoing infusion for quite some time, where you're basically sitting and get this drug intravenously infused. Now, what we try to do is basically designing microrobots as drug delivery vehicles. And that can basically take the drug that might be packaged in nanoliposomes. So these are nanoscale lipid balls in that sense. And lipids are also, it's the main component of our cell membranes, right? So we make synthetic forms thereof. So this is a way we already package some drugs. In fact, how also mRNA-based vaccines are delivered, right? So we have these already nice little nanoballs, that can encapsulate the drug. Now, how do we get them effectively to a target site, for example, to cancerous tissue? So if we package them now onto a microrobot that can be such a hydrogel with magnetic particles inside, or I mentioned before bioinspiration to use kind of robots that have similar locomotion like bacteria. Actually, what we've been working on was also working on biohybrid microrobots. So using real bacteria as a vehicle and leveraging some of the intelligence that nature has already presented us with, basically, but then add external control by working with magnetically responsive bacteria, by adding magnetic material to make them responsive. So there are some that are naturally magnetic. And then we can externally control them with magnetic fields, right?
Yeah.
So this is then basically a way how we try to guide them then to a target location. Now, when I say guiding, I don't mean that upon intravenous injection, we would follow our microrobots and guide them to a location, because actually the blood flow is very effective in distributing them, right? So they are within fractions of a second dispersed and go through the circulation. Now, what we found is that actually by applying rotational magnetic fields, the microrobots we work with, we design them in a way that they can very well turn upon exposure to a rotational magnetic field. So they have a long axis, they have a strong, in that sense, like a little compass needle that would orient with the Earth magnetic field. We have basically created them in a way that they can very well follow changes of the magnetic field. And so they would start rolling and tumbling. And if this is in small vessels, as we come into circular tube, we get smaller and smaller, and the capillaries are then only about 10 micrometers, right? So this is a tenth of a hair, the width of a hair. That we basically managed to get these microrobots to roll along vessel walls and that they can actually get out and enrich tissue, because the difficulty is basically get them out of circulation at locations where you want to have them out, right? A lot, of course, gets right away cleared into the liver. We have the immune system taking care of any such foreign entities in the body. But in this way, we can basically, when we inject and directly apply these rotational magnetic fields, we showed in vitro and then also in small animal studies that we can enrich the target tissue and help basically these microrobots getting out of the vessels and penetrating cancerous tissue more deeply and then delivering their drug there.
Amazing. Yeah, really, really quite sci-fi. It's fantastic. So how do they know when they're in the right place?
Oh, actually, they don't need to know here because we focus our rotational magnetic field to a target location, right? So
Right.
we, of course, need to know where, for example, cancerous tissue is. So for
Yeah.
me, the scenario I would envision in clinics is really we have a scan before of a patient, like, for example, a PET scan, positron emission tomography, where you very nicely can actually resolve also metastatic sites. And then we would basically, upon intravenous injection, scan through these areas with our rotational magnetic field and apply this to help the extravasation locally.
Okay. Yeah, amazing. And another theme of your research is kind of diagnostics. So how are you applying these kinds of ideas to detecting and diagnosing illness?
Yeah. So here, and again, for some, what I'm describing might not sound like a micro robot. To me, it does because it follows these principles of input processing and output. But for
Hmm.
example, we were quite interested in working with ultrasound of a way to communicate with micro robots in vivo, right? Because ultrasound is actually a low-cost point of care diagnostic that has already great use, but also great potential beyond basically giving tissue contrast information, but also to give more molecular information. And that can be
Okay.
done by designing micro robots that can report, for example, activity of certain enzymes that are associated with a certain disease through a change
Mm,
in the acoustic signal they're giving us.
okay.
And so how we could envision this is some of you might know about some ultrasound contrast agents. These are actually gas-filled little microbubbles that are already used
Mm,
in clinics. This especially can help to visualize blood flow because they're intravenously injected, and they swirl around and they give us a very, very strong, bright contrast that has to deal with the interface and differences in acoustic impedance. And basically, we engineered now such microbubbles in a way that um, the mechanical properties of the shell is altered when there is a certain enzyme of interest active, because this enzyme interacts with the shell, and that changes then basically the way it sends back the acoustic signal or the nonlinear backscattering in that sense.
okay. That's really interesting. So obviously that's for diseases that you already have some idea how they function. You know that a particular
Mm.
enzyme is involved. Is there scope to use this kind of technology for researching diseases that are less well understood?
Yes, absolutely. And I think I was actually kind of mentioning this already a bit in your earlier question, but
Hmm.
that there are really many applications outside of the body. And that is to, interact with cells, but interact also on a tissue level and interact also in disease models, right? So, for example, one of our projects actually dealt with understanding some of the mechanical cues involved in breast cancer metastasis. We
Okay.
know how important mechanical forces are in actually our development, but also how changes are associated also with diseases, right? We know we can feel breast cancer because of the fibrotic tissue that gets we get hard clumps. So
Hmm.
you can feel that, and the mechanical changes in the environment also triggers certain pathways in cells. And so we were
Hmm.
trying to understand this better by using, in the end, again, little magnetic microprobes that were embedded in tissue models where we had basically breast cancer cells as a spheroid. So basically, we would create little mini tumors out of established breast cancer cell lines, but
Okay.
embed them in collagen as a constitutive tissue surrounding, but have then
Hmm.
implanted in this surrounding tissue little magnetic microprobes that could perturb and kind of generate forces on this little mini tumor. But also, we could feel also the stiffness basically by seeing how they are affected if we apply controlled external magnetic forces on them. And so we can, on the one hand, understand the mechanical changes, but also apply mechanical forces and then study the behavior.
Like an internal breast exam.
Yeah, yeah, in a way.
And so in 2014, you co founded a spin off company called MagnebotiX that specializes in wireless micromanipulation. So tell me a bit about that company and then the applications that you're kind of targeting there.
Yeah. So one of the core systems is actually an electromagnet that you can integrate into microscopes, right? So it
Okay.
gives means to biologists or other roboticists or anyone who's basically interested in manipulating at a small scale wirelessly. So especially some people that build microrobots want to understand their motion, right? If they are magnetic, so their motion under external magnetic fields. But also you can, for example, there's one interesting field also in drug discovery where proteins need to be crystallized and then harvested, and we need to manipulate these very fragile crystals in a gentle way. And so by having actually soft little micro robots that can be moved, there was a way of also manipulating such crystals. This was one application we also explored. And but then again, also, of course, for single cell studies, for tissue studies, then also in tissue fabrication, where you basically need to generate precisely external fields and magnetic fields and gradients. The system can be used. And we also offer, other variations thereof that you can actually also do preclinical studies on an animal level to test these microrobots that are intended actually for use as, for example, therapeutics in the body.
It's such a fascinating area of research. Applications beyond what I had even imagined. So it must be a really interesting area to work in.
Absolutely, yeah. I feel very blessed and fortunate to have a very enthusiastic team around me, and there are also problems I'm working on today I would have never dreamt of, and who knows what else we're going to work on. Absolutely,
yeah. Yeah, I will be watching your work with interest over the years. Simone, it has been great speaking to you today. I've been chatting to Simone Schuerle, an assistant professor at ETH Zurich. Thanks, Simone.
Thank you, Claire.
Thanks for listening. If you enjoy Robot Talk, please do share the podcast, subscribe, and leave a review to let us know what you think. And make sure you check out our social media channels to see a photo of a magnetically controllable, bacteria-based microrobot that Simone and I talked about. We're at Robot Talk Pod on all the socials. Make sure you tune in next week to hear the recording of our live event on robotic prosthetics and human augmentation. Until then, I've been Claire Asher, and this has been Robot Talk. Robot Talk is brought to you by the Hamlin Centre, Imperial College London.